Something moves in the darkness. Not a sound. Not a flash of movement visible to the eye. A channel catfish, 30 inches of muscle and instinct, turns 20 feet below the surface. Its barb will shift. Its lateral line registers a tremor. Something has entered its world. It has not seen anything. It has not heard anything, not in any way a human would recognize as hearing. But it knows. It already knows. Every angler knows the feeling. The line goes still. The water gives nothing away. And then, without warning, without any signal a human nervous system can process, the fish strikes. For centuries, that mystery was the whole game. The best fishermen on the Mississippi Delta, the Atchafalaya Basin, the rivers of the Deep South, they had their theories. Age bait. Certain smells. The way a lure moved. They passed this knowledge from father to child the way any tradition survives, through practice, observation, and faith in what works. What they did not have was a laboratory. What they did not know was that they were right, almost exactly right, about nearly all of it. The science would take another century to catch up. Fish are not passive creatures waiting to be fooled. They are sensory machines, built by hundreds of millions of years of evolution to perceive a world that human beings can barely imagine. They smell at concentrations that would be invisible to any instrument available a century ago. They feel vibrations from objects they cannot see. They read color through photoreceptors tuned to the specific frequencies that matter most in their underwater world. This is the story of those senses. How they work. What the science actually says. And how the anglers of America's great rivers and lakes, long before any of this was formally understood, discovered through tradition and trial what researchers in university laboratories would spend decades trying to prove. From the extraordinary chemical detection of the channel catfish to the precisely calibrated color vision of the largemouth bass, from the invisible pressure reading system that runs the length of every fish in American waters to the deep-rooted southern bait traditions that encoded this science long before it had a name. This is how fish actually sense the world. Hello my friends, welcome to American Fishing Atlas. Subscribe to the channel, share and like. Thank you. Consider, for a moment, what it means to smell underwater. On land, the sense of smell is a matter of airborne molecules reaching receptor cells, a relatively crude, directional process. In water, chemistry dissolves and disperses. Molecules diffuse outward in every direction, diluting with each foot of travel. By the time a scent has traveled 100 yards through a river, its concentration is vanishingly small. For most animals, that trail is gone. For the channel catfish, it is only beginning. The olfactory system of Ictalurus punctatus, the channel catfish, the most widespread catfish species in the United States, is among the most sensitive chemoreceptor systems known in the vertebrate world. Where a human nose has relatively few olfactory folds, the channel catfish possesses more than 140 folds in its olfactory rosette, a dense, elaborate surface covered with specialized receptor neurons, each tuned to detect specific molecular signatures in the water column. Research conducted at Louisiana State University and confirmed by multiple electrophysiological studies documents the scale of this sensitivity. Channel catfish can detect certain amino acids, the basic molecular building blocks of all protein, at concentrations of less than 2/10,000ths of a part per million. To put that in terms a river fisherman might appreciate, 1 teaspoon of the right chemical, dissolved in a 1-acre pond averaging 3 feet deep, is enough for a catfish to register a response. Some researchers have documented detection thresholds approaching 1 part per 10 billion, a level of chemical acuity that rivals the legendary olfactory powers of bloodhound dogs. But olfaction is only the beginning of the catfish's chemical world. The same fish that smells with astonishing acuity is also, In the words of Dr. John Caprio of Louisiana State University, essentially a swimming tongue. Taste buds in most vertebrates are confined to the mouth. In catfish, they cover the entire body surface, the barbels, the flanks, the fins, even the tail. Ictalurus punctatus can detect certain amino acids through its gustatory system at thresholds 10 to 100 times more sensitive than through olfaction alone. Concentrations measured in the range of 1/10,000,000th of a molar solution. The barbels, those distinctive whisker-like appendages that give catfish their common name, are particularly dense with taste receptors. As a catfish moves through murky water, its barbels are not simply feeling the substrate. They are tasting it, tracking the molecular residue left by prey items that passed through minutes or even hours before. The catfish does not so much find its food as read the water for the story of where food has been. The catfish fishermen of the Mississippi River Delta did not know the words amino acids or chemoreceptor threshold. They did not need to. For generations, stretching back through the post-Civil War era and into the centuries before European contact, when indigenous peoples of the southeastern river systems first discovered the catfish's extraordinary responsiveness to organic compounds in the water, the practitioners of this tradition built their knowledge through observation. Aged bait outperformed fresh bait. Chicken liver, ripened in summer heat, drew fish from farther than anything fresh. Certain fermented mixtures, jealously guarded, passed down as family secrets, worked in ways that could not quite be explained but were demonstrated trip after trip. What these generations of anglers had inadvertently done was optimize their bait for the catfish's actual sensory system. The aging process, fermentation and partial decomposition, breaks proteins down into their component amino acids. The very molecules that catfish olfactory receptors are most exquisitely sensitive to are released in abundance by exactly the kinds of decomposing organic baits that Southern tradition valorized. The science confirmed the folklore. The folklore had been encoding the science for 100 years before anyone ran the experiments. Light behaves differently in water than in air. Long wavelengths, the reds and oranges visible to human eyes, are absorbed within the first few feet of the water column. Shorter wavelengths, blues and greens, penetrate deeper. In stained, tannic southern lakes, even those penetrate poorly. In turbid rivers, visibility measured in inches replaces visibility measured in yards. The eyes of freshwater fish have evolved accordingly. But the adaptations are not what most anglers assume, and the science that revealed them changed the way serious bass fishermen think about color selection. The first scientific study of largemouth bass color vision dates to 1937. A researcher at the Illinois Natural History Survey named Frank Brown trained bass to approach different colored targets using food rewards and documented that the fish could distinguish red from green, a finding remarkable for its time, published when most bass fishermen were still using split cane rods and wooden lures hand-carved in kitchen workshops. 8 decades of subsequent research confirmed and refined that early work. The clearest picture emerged from a 2019 study from researchers at Cornell University and the University of Illinois, who measured the spectral sensitivity of largemouth bass photoreceptors directly. The findings were precise. The largemouth bass, Micropterus samoids, possesses dichromatic color vision, 2 types of cone cells rather than the 3 that give humans their trichromatic perception. One type is maximally sensitive at approximately 535 nanometers, the green portion of the visible spectrum. The other peaks at 614 nanometers, a red-orange wavelength. Rods, the cells responsible for low light and motion detection, peak at around 528 nanometers. The practical implications of this architecture are significant. Bass can distinguish red from virtually any other stimulus with high reliability. Green is also resolved clearly. But the color space between blue and black collapses into near indistinguishability. Chartreuse, a lure color that has inspired decades of debate among bass anglers, registers to the bass eye as essentially white, not the vivid fluorescent signal it appears to a human looking at the same lure in sunlight. The practical takeaways from bass vision research resist simple reduction to lure color selection, because water itself is not a constant. In clear highland reservoirs, where light penetrates to depth without significant filtration, color becomes meaningful down to 15 or 20 feet. In the dark, tannic lakes of the southeast, where cypress trees stain the water with tannins and the effective visibility may be measured in 18 inches, color becomes secondary to contrast, to profile, to vibration. What the research clarifies is the priority structure. Red and green are the colors bass can actively distinguish. Contrast between light and dark, a dark-backed lure against a bright belly, a shad pattern crankbait in silver and white, triggers the shape recognition that is fundamental to predatory behavior. And at close range, when the fish has already oriented to a target through other senses, color functions as a final confirmation trigger. This is not how lure color is sold. It is, however, how it works. Interestingly, the long-standing angler preference for red accented lures, red hooks, red trailers, red-tipped cranks, has accumulated across the same span of time that bass vision research has been quietly establishing the primacy of red in the bass's perceptual world. The conventional explanation has always been intuitive. Red resembles blood, injury, a wounded baitfish. And that explanation is not wrong. What it misses is the neurological specificity, that red is not merely associated with vulnerability, but is the single color that bass eyes are architecturally designed to detect with clarity against all achromatic backgrounds. It is not that red looks like something edible. It is that red is the color bass can actually see. Once again, the tradition preceded the science. Once again, the science explains the tradition. There is no adequate human analogy for the lateral line. It is not hearing, though it shares structural elements with the inner ear. It is not touch, though it responds to physical displacement. It detects the movement of water itself. Pressure gradients, particle velocity, the hydrodynamic signature left in the water column by anything that has recently passed through it. The lateral line of a predatory fish is, in the most accurate sense, a sixth sense, one that has no counterpart in human neurology. The system is ancient. It appears in fish lineages that diverged more than 400 million years ago, which means the lateral line predates the emergence of land vertebrates by roughly 150 million years. It is not a refinement or addition to an existing sensory system. It is foundational to what it means to be a fish. The mechanism operates through structures called neuromasts, clusters of hair cells embedded in the skin, either exposed directly on the surface or running through fluid-filled canals just beneath it. Each hair cell bears tiny cilia that deflect in response to water movement. That deflection generates an electrical signal that travels to the brain. By integrating input from hundreds or thousands of neuromasts distributed across its body, a fish constructs a continuous, real-time map of water movement in its immediate surroundings. The lateral line is tuned, by evolutionary design, to detect the low-frequency vibrations produced by living prey. Frequencies in the range of 1 to 80 cycles per second correspond closely to the tailbeat frequencies of baitfish, shad, minnows, shiners, the small forage that forms the base of the predatory fish's diet. A largemouth bass positioned in structure can detect the pressure signature of a school of shad moving 40 to 50 feet away, not through its ears, but through this system. The implications for predatory behavior are profound. Blinded bass, in controlled research settings, remain capable of locating and striking prey with reasonable accuracy. But when the lateral line is chemically suppressed, when cobalt ions are applied to block neuromass function, the same blinded bass can no longer hunt effectively. Vision, it turns out, is optional. The lateral line is not. The system is also directional with extraordinary precision. A feeding base does not merely detect the general presence of vibration. It can distinguish the hydrodynamic trail of a single fleeing minnow from the ambient turbulence of a river pool. Researchers have documented the ability of predatory fish to discriminate between species-specific vibration patterns. The signature of a crayfish versus the signature of a shiner, registered through nothing more than the geometry of water displacement. Sound travels roughly 4 times faster in water than in air. Fish have evolved to exploit this advantage through 2 overlapping systems: the lateral line for low-frequency near-field vibration and the inner ear for higher-frequency, far-field sound. In catfish, and in related species including carp and minnows, an additional anatomical structure dramatically extends this hearing range. The Weberian apparatus is a series of small interlocking bones, derived from modified vertebrae, that physically connect the swim bladder to the inner ear. The swim bladder, a gas-filled organ used for buoyancy regulation, functions in this configuration as an acoustic amplifier. It receives sound pressure waves and transmits them mechanically to the auditory system. The result is a hearing range that extends to significantly higher frequencies than most other freshwater fish can detect, and a sensitivity to pressure changes that includes not just conventional sound, but the low-frequency seismic movements transmitted through water, and even into the substrate of the riverbed. Catfish can detect the movement of organisms walking along the bank above water level. Fishermen who know this walk softly. They always have. The moment a predatory fish commits to a strike is not a single event. It is the convergence of multiple sensory inputs arriving in rapid sequence. At distance, say 40 feet, the lateral line of a base registers a low-frequency vibration from a moving lure. The fish orients toward the source. As it closes the distance, the vibration signal strengthens, and the visual system begins to contribute. Shape, contrast, the movement pattern of the lure against available light. At 10 to 15 feet, the fish has assembled enough sensory data to make a commitment decision. At 5 feet or less, the lateral line system takes over in its most precise mode, close-range hydrodynamic discrimination, confirming that the object in question matches the profile of something edible. The strike itself is explosive, not because fish are impulsive, but because they are certain. The sensory calculation has been running since the lure entered the fish's world, and by the time the bass commits, the conclusion is not a guess. It is a verdict. There is a particular kind of knowledge that resists translation into language, knowledge that lives in the hands, in the rhythm of a cast, in the instinct to add a day's aging to bait before a river trip. The catfish fishermen of Louisiana and Mississippi who developed their stink bait traditions did not document their reasoning. They did not need to. The results were self-documenting, generation after generation. What is now understood is that this traditional knowledge was tracking something real in the biological world. The amino acid sensitivity of catfish olfactory receptors is not a laboratory abstraction. It is the specific mechanism that Southern bait tradition was unknowingly exploiting. The catfish's full-body chemical detection system is not a curiosity from a biology textbook. It is the reason that certain baits work where others fail, in certain water temperatures and current conditions, with a specificity that once seemed mystical. Knowledge and science are not always in opposition. Sometimes they are converging paths to the same truth. Much about fish sensory systems remains genuinely unresolved. The lateral line's precise discrimination capabilities, its ability to distinguish species-specific hydrodynamic signatures, to track wake trails through turbulent water, to function simultaneously across hundreds of independent neuromasts, is an area of active research. Artificial lateral line systems, modeled on fish neuromast architecture, are currently being developed for underwater robotics and sonar applications. Electroreception, the ability of certain fish species to detect weak electrical fields, remains perhaps the least understood of all fish senses. While best known for marine species, like sharks, freshwater electroreception exists in catfish lineages as well, allowing the detection of the bioelectric fields generated by the muscle contractions of prey. The full ecological role of this capacity in American freshwater species is still being mapped. The chemistry of fish olfaction continues to reveal surprises. Recent research has documented that fish can detect and respond to alarm substances, molecular signals released by injured fish of the same species, and that these chemical cues can alter feeding behavior, schooling patterns, and habitat preference in ways that cascade through entire fishery ecosystems. The implications for fish population dynamics and for the management of American sport fishing resources are only beginning to be understood. On the Mississippi, on the rivers of the Deep South, on the highland reservoirs of Tennessee, and the glacial lakes of the upper Midwest, the fish are running their calculations constantly. Every movement in the water column is registered and assessed. Every molecule dissolved in the current is cataloged against a receptor library of extraordinary sensitivity. Every shift in light and shadow is processed through a visual system tuned not to beauty but to survival. The fisherman on the bank does not know any of this. The fisherman on the bank drops a handline baited with ripened chicken liver into a dark pool and waits. He learned to do this from his father. His father learned it from his. The reason it works was never explained. It never needed to be. The catfish, 30 feet below, registers a molecule, turns, begins to move. The science is new. The knowing is old. The river does not distinguish between them.